Hi guys, welcome back. After your interlude with Dr. Carr, it's sort of good to be with you again. Hopefully, through those lectures, you've now had a really good exposure to some of the basic properties of the um, uh, fatty acid and other molecules that make up biological membranes, um, some of their physical properties, and some of the ways in which the proteins that interact with or embedded in those membranes behave, and some of the distinctions between that behavior and some of the behavior we saw when we first talked about protein structure and folding at the beginning of the course. And those different physical properties, the sort of phase separated behavior of these membranes, is absolutely critical to a huge range of basic biology, but it's key to the last few steps we're going to take through metabolism, because that diffusive barrier that those membranes create and the way in which those membrane embedded proteins behave is key to this last step of the process in how we extract useful energy from this oxidative cascade that we've been building up as we came through glycolysis and TCA cycle, um, to these high potential electrons we now want to turn into ATP, right? Okay, and the whole thing starts with what's essentially going to be, as I mentioned before, the construction of an electrical wire that is embedded in a particular membrane in a living cell, and then coupling the flow of those electrons down that wire to the physical work necessary to drive synthesis of ATP. All right, so just to sort of remind you guys again of where we were, right? Remember thinking about the TCA cycle, and again, just to re-reference you guys, TCA cycle, as you recall, is where and if we have oxygen available, is where the largest possible flux of NADH is going to be created as a result of TCA cycle. And as you hopefully recall, TCA cycle is, of course, running here in the matrix of the mitochondria. And just to remind you guys a little bit of some of the basic properties of the mitochondria, in terms of the basic structure, because we need to know something about this with respect to what's going on here. Mitochondria, as you again recall, is this double membraned endosymbiotic remnant of a alpha gram-negative alpha proteobacter that some ancestor archaeal of eukaryotes engulfed once upon a time and didn't destroy, but turned into a little energy factory. Has this outer membrane, has an inner membrane, and that inner membrane is then folded into the sort of invaginations, which are known as cristae. And so we have two sort of like soluble spaces, lumens, if you want to think about it that way. One between the outer membrane and the inner membrane, called the intermembrane space, and then this sort of volume on the inside of the inner membrane in the sort of between these folded cristae, that would be known as, of course, the matrix. And as you guys, of course, recall, that's where TCA cycle is running, cranking out all this NADH. Okay, now the really key thing here is that this electron wire that we're going to be talking about, these protein complexes that let us take all those high potential electrons and turn them into ATP, is embedded in that membrane, that inner mitochondrial membrane, right? And what we're going to end up doing here, of course, is we're going to take that set of electrons and we're going to run them through these protein complexes, and those electrons are going to flow down the different these different complexes into ultimately the receiving sink of oxygen. We're going to create water out of that, right? And in the process of doing this, we're going to create the conditions that let us turn that flow of electrons into energy. All right. Now, before we go there, just make one additional sort of little comment here about something. We're sort of used to thinking about mitochondria, right? Of course, and the way it's always drawn, that picture I just showed you, as this dainty little sort of double membrane thing with these nice little fragile folded cristae, right? And here's an example of a liver, the mammalian liver mitochondrion. You see those two outer membranes here, and it's sort of, this is a mitochondrion that's been turned on its end and then sliced for some negative stain electron microscopy. And so what you're seeing is sort of the projection of those cristae sliced transversely, and in the holes in the middle there, right? You're sort of seeing those inner mitochondrial invaginations, right? Of the inner mitochondrial membrane, right? and the matrix out here, right? And the intermembrane space in this case seems kind of thin, right? It's light here. You can see it in the sort of inner and outer membrane around the edges here. Okay, the important thing is, and that's the way it's always drawn, that's a resting mitochondria. That's a mitochondria that's not undergoing really active driving mitochondrial respiration. Because when a mitochondria engages in full-on electron transport and respiration, aerobic respiration, it doesn't look like that. They change, right? And they change in a dramatic way. And this is an example of that. This is that same sort of liver mitochondria under actively respiring. And notice something pretty dramatic. The inner mitochondrial membrane condenses, it shrinks down and becomes much more sort of collapsed. The intermembrane space gets dramatically more dense. I'm sorry, the matrix gets dramatically more dense. The intermembrane space sort of expands, becomes much more sort of larger fraction of the total volume. And there's not as nearly as much protein content stuff in there. So it's in these negative staining EMs, it looks almost empty. It's not, but it's much more fluid filled, much more dilute than the matrix. This change in morphology is critical to the rearrangement of these protein complexes into an efficient system, this efficient wire that's going to drive production of ATP, right? And this rearrangement is key to that process happening as efficient as possible. Okay, so remember, these complexes, these protein complexes we're going to talk about, are embedded on inside this inner mitochondrial membrane, and sort of just in 35,000 foot view, what we're looking at for this electron transport process, we're just going to take the 4 protein complexes, 2 diffusible carriers, one which is membrane soluble and one which is going to surf along the outer surface of the inner mitochondrial membrane, and as well as 5 different types of electron carriers and 2 different kinds of metals, right? So this is sort of like an interesting sort of collision point in multiple different ways between sort of redox chemistry that I was alluding to back before we took your little excursion into membrane biology, and the sort of membrane biology of how we get this all to work, right? All right, so 35,000 foot view. Remember, again, we have that NADH that we're making through TCA cycle, or that we've shifted equivalents into the mitochondria from glycolysis. If we want to sort of run those through this as well. The first transfer is going to, of course, take those NADH equivalents and transfer them to the first complex known as NADHQ oxidoreductase, or more simply, just complex 1, right? Notice something here, right? That the standard reduction potential of complex 1 is still relatively reducing, but it's oxidizing relative to NADH, if you remember where the NADH redox potential was sitting. So this transfer is going to happen reasonably efficiently. And what you're going to see is that this redox potential, as you go down the chain, gets more and more and more oxidizing until we get all the way to oxygen. Okay. The complex 1, or NADHQ oxidoreductase, it transfers those electrons to the first diffusible carrier, coenzyme Q. This is the only electron carrier, once we get out of the inputs, NADH and FADH2, that does not involve a metal, right? All the rest of our transport steps are going to involve metals, but this one doesn't. This carrier, as you're going to see, is a little aromatic diketone that sucks those electrons out of complex 1. It's that reduction potential is getting more oxidizing. So you can sort of see how this potential flow is going to be going in the direction of more reducing to more oxidizing. Now, another important thing is the electron transport chain. It's called an electron transport chain, but it's bifurcated right at the beginning. There are 2 different inputs into this coenzyme Q pool. One is from complex 1. The other is from so-called complex 2, which is so-called succinate Q reductase. You've seen this protein before. We've talked about it a little bit, and we're going to talk about it again, but it directly feeds the equivalents from the FADH2 production out of TCA cycle straight into the coenzyme Q pool. All right, those electrons then are transferred from coenzyme Q to complex 3, or Q cytochrome C oxidoreductase. Notice our redox potential is flowing towards more positive. The electrons from complex 3 then move to our second diffusible carrier, cytochrome C, which ultimately transfers its electrons to complex 4, or cytochrome C oxidase. which has a significantly more positive or standard reduction potential, so it's significantly more oxidizing than we had up here. So again, that sort of downhill potential energy flow makes a lot of sense in terms of how these standard reduction potentials are stacked up. And then cytochrome C oxidase, complex 4, is going to deliver those electrons to oxygen, ultimately resulting in the production of water. Okay, so the name of the game here, the purpose of this electron transport chain, you can sort of think about in sort of two ways, big picture, right? So again, you got TCA cycle going, cranking out NADH, FADH2. We want this process to keep going so that we can make more ATP, or we can supply these intermediates, right, that we need for doing biosynthesis, right? We've got to regenerate the NAD plus and the FADH pools, or the FAD pools, right? So just like we saw with GAPDH, one of the goals of this process is to dump these reducing equivalents into oxygen so we can regenerate those oxidizing equivalents we need to drive the chemistry of the TCA cycle. So transferring those electrons is part and parcel of regenerating those oxidizing equivalents. The second big goal of the electron transport is to couple that process of this electron flow to the movement of protons, the directional movement of protons out of the mitochondrial matrix and into the intermembrane space, right? So essentially, we're running an electric wire whose principal, or one of its principal goals, is to act as a proton pump. We're going to use that electron flow down that oxidizing cascade to drive protons, pump protons across the intermitochondrial membrane out of the matrix, right? And it's that potential energy difference between the protons in the intermembrane space and the matrix we're then going to couple to in the last step of this process to make ATP. Okay, now turns out proteins are really bad at transporting electrons, right? And that's really what we need to do here. But the amino acids, standard amino acid side chains, really are not very good at this. I mean, you can, like we've seen, you can kind of coax cysteine into it, kind of, sort of, a little bit, through the oxidation shift of thiol to a disulfide. But, you know, otherwise, and that's not even that great, proteins just don't do this very well, right? So the process in every case within these protein complexes is going to involve a metal of some kind, right? Iron or copper in one form or another. And we're going to look at that in some detail as we go along here. So again, the idea is we're going to take this NADH, or we're going to take the FADH2, and we're going to flow them through a set of metal-based electron carriers, initially into an organic-based carrier, which will then pump them back into complexes, again using metals, to transfer them all the way ultimately into making water, right? So in the next video, we're going to take up a look at how this works with this first complex, and think a little bit about this process of electron transfer and proton pumping in sort of a big picture sense, to think about how this might be working.